My name is Callum. I am a marine scientist at the Letchworth Marine Biolabs, part of the Marine Biolabs and Surveys team. I'm here to take you through kind of a day in the life, a very brief day in the life of what we do at the lab itself. So come along and see what we do, and I'll walk you through everything. This little video just shows some of our samples in storage. This storage area contains a large number of samples that are either waiting to be processed or they have been processed and they're just waiting for external QC. This is basically where we go and collect our samples in order to go and process them in the washroom afterwards. They're separated from the actual lab itself and they are just stored here until we need them. Probably the most important thing that we do in the lab is sieving. This is the first step in any sample processing procedure, and it's what we do most of the time. We get the samples in and we separate them into different fractions. We separate them from 4 mil, 2 mil, 1 mil, and then half mil. Depending on the project, we either use the half mil or the 1 mil as the final fraction. But as you can see here, this is the sieving process. This is what we do. You take the sample, you decant the formalin, the preservative that we use, and then we wash the sample out with water and put it through the different fractions themselves. Once we've done that, we then separate them into trays, or the 2 and the 4 mil we separate into trays, and then the 1 mil and the half mil we put into pots, which we then take back to our benches where we then pick through the microscope where it's easier. If a sample is really sedimenty, what we do is we take a small portion of the sediment, we blast that to suspend any animals in the sediment into the water, which we then decant off and we can easily pick through, keeping a bit left just to make sure that we haven't missed anything. The most time-consuming process of our day has to be the picking process. We're going through the samples basically bit by bit. As you can see, there are rings on my petri dish there just to help me work through the sample and picking out all the animals that we see. Sometimes we can pick only the ones that are full, so that we only have heads to make it easier for us to ID. Sometimes we have to pick every part of the animal. It depends whether or not we are biomassing for that particular project. Now, just coming up now are going to be some of the animals that we do find in the samples. All it makes quite arduous and tiring work sometimes. Really cool when you actually finally get to see the animals and actually look at them properly under the microscope. So just coming up here, we just got a few examples of some of the animals that we find in the actual picking process. But each sample is then separated either into taxonomic groups, species, placed into vials. See that I picked up a worm, and then moved on to the ID process. So this is the ID process. We've got the sample there having a look at, split into this different species that we find in and around our sample area. Each sample is then written down onto a sheet of paper, which is then entered into our database. You can see there that we look at all different features depending on the animal itself. We're looking at various features just to ID it. Sometimes the animal is so small that we actually need to take it to a different microscope, and that is further magnification, allowing us to look at more detail on the animal, such as particular hairs or chaetae. And then we use the ID books that we can see there in order to help us. So we use keys through the books, and then we can ID the animal a lot easier. Like I said, this is probably the most time-consuming task, the picking and the ID process. But once it's completed, it then goes back to a taxonomic specialist who then QCs it inside the lab, making sure that we've got the best and correct data we can for that project. Another time-consuming part is the biomassing. Biomassing takes quite a while due to the size of the animals. Sometimes trying to put them onto a scale is quite hard, and waiting for things in order to settle on the scale so you can read a number. The reason why we do that is certain projects require it for the reporting and also for statistical analysis. So it's just something that we do. Again, here is an example of a video of how we do it, and it's always good to have a good podcast or listen to some good music while we do it. Last thing I wanted to talk about is the part where we actually get out of the lab and go onshore and offshore when we go on survey. Now, sometimes these surveys come from a wide range of different specifications, whether we go on boats, whereas we're just walking along a shore, and sometimes we only take videos, sometimes we take samples and bring them back to the lab as well, but at least we get to go outside and enjoy the animals in their natural habitat as well as seeing them in the lab as well. And surveys are now ramping up. We're doing a lot more survey work now, which is really good fun. And if you really enjoy going outside, it's incredible. Just spending hours outside working away. It can be hard, it can be tough sometimes, but at the end of the day, you're outside and you can't really complain about that. So I've got some photos here just showing and highlighting what we actually do on survey work. Some grab images and also some really cool training opportunities that we get in order to go on these surveys as well. But I will leave you with those photos, and I just want to say thanks so much for watching. I hope it's been an informative video, just giving you a highlight of what we kind of do at the Biolabs. And if you have any other questions, feel free to get in touch. But thanks so much. Bye bye.